Good afternoon. Today I will teach Invic Test, which is used to differentiate E. coli from the Enterobacter aerogenes. Invic Test consists of the set of four tests. The first test is Indole Test. So this Invic Test, it is the test used to differentiate fecal coliforms which are E. coli, and this particular test differentiates E. coli from Enterobacter. That these are the non-fecal coliforms. For differentiation of E. coli from Enterobacter, Invic test is used, which consists of four subtests. In Invic, I stands for Indole test, M stands for Methyl red test, V stands for Vogus Prosker test, and C stands for Citrate utilization test. So we have to perform these four tests for differentiation of E. coli from Enterobacter aerogenes. In Indole test, different media for all four tests are used. We'll see Indole test. The medium used for Indole test is 1% tryptone or 2% peptone water. Peptone and tryptone in the medium supplies tryptophan, that is the amino acid, and the organisms having tryptophanase enzyme along with specific transfer system can produce indole from tryptophan. So we have to check the presence of tryptophanase enzyme and production of indole in this particular test. So indole is a volatile in nature and extracted on addition of xylene, which forms a ring on the top of the medium as the indole is volatile. Then indole is detected colorimetrically by addition of Kovacs reagent. This Kovacs reagent is nothing but the paradimethyl aminobenzaldehyde, that is the organic chemical compound, and paradimethyl aminobenzaldehyde from Kovacs reagent reacts with indole and forms a pink ring on the surface of the medium. So this is the photograph of indole test. At the left side you can see the positive indole test, and at the right side the negative indole test. The pink ring at the surface of the medium you can see, which is the positive indole test. Okay. Then the second test is methyl red test, in which the methyl red indicator is used. The medium used for this test is glucose phosphate broth, that is GPB. In some organisms, glucose is broken down by mixed acid fermentation into lactic acid, succinic acid, water and CO2, while some organisms produce the neutral compounds like butanediol or acetyl methyl carbinol. For example, Enterobacter can produce such compounds, and E. coli utilizes glucose very rapidly and produce acid, while Enterobacter uses glucose slowly and therefore does not produce acid after incubation in the tube of glucose phosphate broth inoculated with the organism. So in this particular test, for detection of acid production like lactic acid and succinic acid, methyl red indicator is used, which is the pH indicator. And the methyl red is red below the pH 4.5, while it is yellow above pH 6, and therefore red color is produced only due to acid production. So by adding methyl red indicator, we can check the production of acid, that is the formation of red color, and test the organism whether it is E. coli, or whether we can say that the methyl red test is positive if the red color is developed after addition of methyl red indicator. This is the photograph of the methyl red test. At the left side you can see the negative methyl red test, in which there may be the production of acetyl methyl carbinol. After addition of methyl red, if the whole tube becomes red, then the test is said to be methyl red test positive. Here one more test in this right side photograph is shown, that is Vogus Prosker test, in which also there is a development of pink color, but the addition of reagents is different for this test. Okay. Now the third test in this set of tests, that is the Vogus Prosker test. The medium used is glucose phosphate broth for this test. Same medium for the methyl red test is used for Vogus Prosker test. The test is named due to the scientist who discovered this test, that is Vogus and Prosker. Many bacteria like Enterobacter produce the compound acetylmethylcarbinone and 2,3-butanediol, which are the neutral compounds from glucose, and these compounds under alkaline conditions reacts with the guanidino group of alpha naphthol and give red color. Okay, so after inoculation of the organism and incubation, this Vogus Prosker test is performed by addition of 40% KOH, that is the potassium hydroxides, and the conditions are made alkaline, and the presence of acetylmethylcarbinol is detected by addition of alpha naphthol. So the guanidino group of alpha naphthol reacts with acetylmethylcarbinol under alkaline condition and gives a red color. So this particular test, Vogus Prosker test, is positive for Enterobacter aerogenes and it is negative for Escherichia coli. So formation of red color we can see in this particular test. This is the positive test, that is the addition of alpha naphthol and 40% KOH. 40% KOH in 3:1 proportion is added, and if the no pink color develops, the VP test is said to be negative. This is the photograph of all 4 tests like Indole test, methyl red test, citrate utilization test and the Vogus Prosker test. So this is the citrate utilization test. So these are the 4 tests shown in this photograph. The 4th test in this Invic test, that is the citrate utilization test. The test is carried out to detect the ability of organism, any organism, to use citrate as a sole carbon source in the medium. So in this particular medium, the Koser citrate medium is used and it contains sodium citrate as a sole source of carbon, and only those organisms having ability to utilize the sodium citrate as a carbon source can grow in this particular medium, because we have not added any carbon source in this medium. So for this test, Koser citrate medium is used, which contains sodium citrate as a sole source of carbon, and the medium is colorless, and the test is said to be positive if turbidity is observed. If the organism utilizes sodium citrate as a sole source of carbon, the organism will grow in this particular medium and the medium will become turbid, and if no turbidity is observed, the test is said to be negative. So in case of Enterobacter aerogenes, turbidity is observed, the citrate utilization test is said to be positive. If no turbidity is observed, the test is said to be negative, and it is negative for the organism E. coli. So this test can be also performed by using another medium, that is the solid medium, instead of Koser citrate medium, in which the blue indicator, that is the bromothymol blue, is added in this medium, and the detection of citrate utilization is detected by change in color of medium from green to blue, and the medium is called as Simon's citrate medium. The citrate source is same, but only in this medium the agar is used to make the medium solid, and the indicator, that is the bromothymol blue indicator, is used, so that in case of positive test, the color change of the medium will be green to blue. Okay. This particular chart shows you how the Invic test differentiates E. coli from Enterobacter. So if we perform Indole test using the tryptone water or peptone water, methyl red using the GPB medium, and VP test using the GPB medium, citrate utilization test using the Koser citrate medium, you can see the results, you can get the results like this. If you get the results like this chart, that is Indole test and MR test positive for E. coli. Enterovector aerogens, for enterovector aerogens, Indole and MR tests are negative, while the VP and Citrate utilization tests are negative for E. coli and positive for Enterobacter aerogenes. So, this particular test is useful in differentiating E. coli from Enterobacter aerogenes. This is one more test used to differentiate or confirm the presence of E. coli in the water sample. That test is known as elevated temperature test, and this test is proposed by Ejekman. So this particular test is also called as Ejekman test. He assumed that only fecal type of coliforms like E. coli could grow at 45.5 degrees centigrade with production of acid and gas. Okay, so the given organism which we have to identify, if it is inoculated in lactose broth medium, that is the liquid medium, and incubated at 45.5 degrees centigrade for 48 hours, and the production of gas is positive result, and that is observed in case of E. coli. If no gas production or acid production, then E. coli is absent. So this particular Ejekman test or elevated temperature test is positive for E. coli, and presence of E. coli indicates fecal pollution of water. So number of tests, so these two tests, elevated temperature test and Invic test, are important tests for the detection of presence of E. coli and differentiation of E. coli from Enterobacter aerogenes. Okay. So today we have seen the